Wonderful. So, welcome everybody again to another episode of the ideaXme show, that show that profiles the humans behind the really big ideas that are shaping our world and inspiring future creators, and for all those that like really great stories. I'm Ira Pastor. I'm your aging, health, and longevity ambassador along for this journey. So, on the last several shows, we've been spending a lot of time on as we refer to, the different hierarchical levels of the architecture of life and aging. We've been talking about the genome, the microbiome, extracellular matrices, and systems biology. However, at the end of the day, while all of these wonderful tools are extremely important in the maintenance of our daily health and wellness, we can't forget that when things go wrong, the trickle down is unfortunately a wide range of chronic degenerative diseases responsible for human suffering and death, and as of now, a global healthcare expenditure of around $7 trillion every year. And of course, one of the major groups of pathologies responsible for a piece of that figure in 2019 remains diabetes, a group of metabolic and endocrinologic pathologies affecting over 350 million patients worldwide, and projected by the World Health Organization to surpass 500 million within the next 2 decades. And the global estimated economic cost of diabetes medical and reduced productivity in 2018 was estimated somewhere between $3 and $4 trillion. Type 1 diabetes, also known as juvenile diabetes, we've talked about before, is a form of diabetes where very little or no insulin is produced by the pancreas, and while its cause is still slightly unknown, it's believed to involve a combination of both genetic and environmental factors, and the underlying mechanisms involve an autoimmune destruction of the insulin-producing beta cells in the pancreas. Complications of diabetes, including retinopathy, nephropathy, neuropathy, and cardiovascular disease, are major causes of morbidity and mortality, as well as the severe hypoglycemia and diabetic ketoacidosis that are associated with high mortality rates as well, especially in young patients. In addition to diet and lifestyle changes and oral hypoglycemic agents, which are reserved more for type 2 diabetes, which is a different topic, the core pharmacotherapeutic intervention for active type 1 disease in 2019 remains replacement injections of insulin, a medication that we can't forget was approved close to 100 years ago in 1922. So, on a past show, we touched on some of the translational work that's going on in the area of exogenous replacement of pancreatic isolates and discussed the topic of stem cells a bit. Today, with our guest, we're going to be exploring an entirely new angle of intervention against this area of substantial unmet medical need, looking at novel technologies for inducing endogenous repair and regeneration of the pancreas via tools for immunomodulation, which may have a wide-reaching implication not just in diabetes, but in a range of other autoimmune disorders and cancer. Dr. Denise Faustman is an MD and a PhD in transplantation immunology. She's Associate Professor of Medicine at Harvard University, Director of the Immunobiology Laboratory at Mass General Hospital, and her work specializes specifically in both diabetes type 1 as well as other autoimmune disorders. And her team is seeking novel and potentially curative treatments based on endogenous regeneration of the pancreas after the autoimmunity process is removed. For the last 20 years, her lab has been a leader investigating the potential of the Bacillus calmette-guerin vaccine, a vaccine primarily used in the past for treatment of tuberculosis and occasionally in bladder cancer, as a tool to prevent and reverse autoimmune diseases such as type 1 diabetes. Her research has advanced from early preclinical work into clinical trials, and recently showed that in 2018, long-term data on human subjects treated with BCG vaccine followed for more than 5 years had durable and significant improvement in HbA1c numbers, potentially a fascinating range of mechanisms involved, including eliminating autoreactive T cells, enhancing regulatory C cells. We'll get into a lot of this. In addition to all of that, the Faustman Lab is also involved, as mentioned, in application of this knowledge in oncology, other autoimmune disorders, which obviously represents another basket of hundreds of millions of patients. All that being said, Dr. Faustman, thank you so much for taking the time to come on the show today.
Well, thank you, Ira. What a great background. There's hardly anything for me to say.
Well, we have a lot more to talk about.
Okay.
So, really appreciate you being here. Typically we start off the show by giving our guests the floor, just a little bit more about you, your background, how you got interested in science, medicine, immunology, and ultimately your path to this rather, I'll say, unconventional but fascinating area of diabetes treatment.
Well, it's always hard to know where to start, but everybody goes like, why did you want to be a doctor? And all I can say is at the age of 5, I thought my pediatrician was too serious, and I thought, the visits should be much more fun, even if you're getting a lot of immunization. So I think it started kind of early. But I got interested in research because of a fabulous program in high school that took high school kids up to the Jackson labs and gave them projects, even in high school, for 3 to 4 months in the summer. And it was the first time somebody gave me true responsibility. You think it out, you do it. And I love the fact that somebody would respect somebody that young and give them the opportunity to think, in kind of a free format. It wasn't a textbook. It wasn't a prescribed answer. And I think that early experience really got me hooked on science, not just being a doctor.
Before we get into some of the work that you're doing now and sort of what you've been doing the last couple of decades, when I look at work like yours, the Nobel Prize winner Barry Marshall comes to mind, who did he did this work and that was just totally it went against sort of the current medical doctrine of, in his case, peptic ulcers and gastric cancer and things of this nature, looking at, a very simple bacterium that, it just wasn't on the radar. Everything was looking in other directions in terms of stress and spicy food and too much acid, but a discovery that ultimately allowed for a breakthrough in a wide range of sort of GI conditions. And I look at your work in, with BCG as sort of being much more fascinating than even that. Can you just tell a little bit before we get into some of the work itself, sort of, I guess, what it's been like for you the last couple decades in really charging in this your own direction, let's say, in an era where everything's been obviously it's been heavily dominated by insulin for the last century.
Yeah, insulin and insulin pumps. Yeah,
right.
We thought, human behavior always surprises me because I think they're trained to follow the data. And at one point in my career, somebody said, Faustman, you follow the data, not the dogma all the time. And I think that's true. I think what I always know is the truth, or the likely truth, is what we've proven the 6 months before, the year before, 10 years before in our lab. So if that's the truth, what we're working on, then we follow in the next line of that truth to get the next answer. And that, truthfully, as you probably are picking up, hasn't always been the what the masses were doing.
Mm-hmm.
And it just repeats itself over and over again. I mean, we thought it was a rather trivial observation when we got done with the first phase 1 trial in BCG that we developed these ultra-sensitive assays for remaining pancreas function using the C-peptide. And all of a sudden, there was this 50-year belief in diabetes that you got diabetes, every medical textbook has it, you have a honeymoon of a year, 2 years, and then your pancreas is dead, okay, if no insulin secretion. So by developing these really fortified C-peptide assays, we all of a sudden started to prove in thousands of subjects that their pancreas lived on for decades. Not always, but the majority lived on for 10 years. Some people 50 years. It wasn't just somebody by histology could see an islet possible sitting in the pancreas. It was that the actual islets were secreting insulin. And when we published that paper, I mean, it's kind of a funny story. Okay, there's the data. And nobody's really paying attention to it because it changes every figure in every medical textbook. And there's a guy in England who was getting very similar data 2 years later, screening urine. He was trying to pick up kids with type 2 diabetes. And I saw him at a meeting. He hugged me and said, I'm so glad to finally meet you. And he goes, well, I have good news and I have bad news. And I said, well, what's the good news? He goes, well, I'm duplicating your data. I'm actually seeing urine collected from kids in the British countryside, and they've got C-peptide in it. And I said, well, that's great. I'm glad to hear you're going to be the first one who replicates it. He goes, yeah, but then there's the bad data. And I said, well, what's the bad data? He says, well, I presented it, at the equivalent of endocrine grand rounds, and the most senior person sitting there at the end stood up, kind of shrugged his shoulders, walked down the auditorium, and said, now we got 2 damn assays that don't work. So I think that story just like epitomizes that. Maybe here's another one from Mark Twain. I'm all in favor of progress, but change I can't stand. So things just go on and on and on, often in the wrong direction. And in science, not the internet, but in science, the amount of volume of things for sea change to translate to the public is very slow and very arduous. And I think it boils down to human behavior.
Mm-hmm.
It is a nice life to keep confirming what all your friends have confirmed. And it's a nice life to go to every meeting year after year, and everybody's sitting and they've made, progress, and it's on the same theme, and you're well liked. What you learn is if you follow a different pathway where you're seeing a different signal, it's uncomfortable for people.
Mm-hmm.
And so I often think about the public pouring so many billions into research. And I think I don't have the solution here. I could just define the problem. We have to have more carrots and rewards for innovation in biology, not just the repeat of the same because it's so safe of a career. Okay?
Absolutely.
And we have an obligation to the people who support these kind of research projects to give them the information earlier, whether it's open access journals or whether it's, it's great you're doing zebrafish, but have you tried it on human cells?
Mm-hmm.
To translate faster and not just have a very comfortable life of doing the same mouse experiment over and over. But that's like a bigger picture outside of my scope of what I can do. But you're right, you're on point. And so, innovators don't necessarily get invited to every cocktail party.
Mm-hmm. Mm-hmm. Absolutely. So, I appreciate it. So setting now that backdrop. So, BCG vaccine, for those unaware, it is a product that I had to look up the date. I mean, I knew it was old, but it came out on the market in 1921, so the year the year before insulin.
Yeah, isn't that ironic?
hit the market. It's still till this day on the World Health Organization's list of essential medicines, where literally tens, if not hundreds of millions of children are given it every year for tuberculosis and a couple other disorders, I believe. Could you talk a little bit? This is a couple-part question here, so take your time with it. Could you talk a little bit about mechanism? Because obviously the immune system, it has its ups, its downs. It's when you get into immunomodulation, immunosuppression, immunosenescence, all these things that are going on, it really, in many ways, is this frontier. I think of it as more, in many ways, more complicated than the brain. There's just so much going on there. If you could talk a little bit about just the general stuff that you're seeing in terms of immunomodulation, what's going on with different cytokines and cells and so forth. And secondarily to that, I would love if you felt like going into the hygiene topic, because
I was thinking that's where you were headed.
Yeah, I'd like to go into that as well, because we've done previous shows with sort of this weird correlation that exists in sort of dirtier areas of the world, or where there's other malaria and other types of infectious diseases where you don't see a lot of the autoimmune disorders. Can you talk a little about the hygiene theme as well, and some of your hypothesis here of why all of this makes sense? And I'll give you the floor for as long as you want on that.
Break it up into, Ira, 2 parts, and one is like, what's the big picture here, the hygiene hypothesis, and then what was kind of our 101 journey to why we got back to BCG. Like, why did we, was it we just picked it off the shelf, or how was it that we identified that as an important vaccine to reintroduce? Because the pathways are a little different. So the 101, what I call the California talk, is like the typical patient comes in and they've got 2 kids with type 1 diabetes or 1 kid with type 1 diabetes, and they go like, why did we get this? Okay, grandmother doesn't have it, Aunt Louise doesn't have it. And like my kids 2 or my kids 5, and I thought it was supposed to occur, at age 20 or 18. And so the hygiene hypothesis, which I think is spot on, first came out of England, around 1960, 65, around there. And they noticed that there was this burgeoning increase in allergies, and then gradually the hygiene hypothesis got applied to autoimmunity. And they went back to do the demographics of who got allergies and autoimmunity. And the demographics were interesting. It was kids of wealthier background, small sample size, never living on a farm, meticulous medical care, with plenty access to antibiotics. And the kids who didn't get autoimmune allergies were farm kids with, 8 kids in the family, limited access to medical care, and on and on. And those demographics continue to this day. So the joke has always been that the northern Scandinavian countries get so much autoimmunity because their houses are too clean. But there's a really pretty serious signal that if you live near the equator and, you live in the dirt and you have parasitic infections and you don't have access to antibiotics and you have to ride out through every infection, that there's no autoimmunity. And then you get to the clean, wealthier households of U.S. and Europe, and you got autoimmunity and allergies. So that's the hygiene hypothesis. And how that kind of ties together with BCG is, as you said, BCG is really a form of mycobacteria. And it's a form of mycobacteria that the story there was it was isolated from the udders of cows. Okay, so this is like a really high-tech drug we're using. And it's kind of the cow version of tuberculosis. So how they developed this vaccine was that people were dying from tuberculosis. It used to be known as consumption, and they realized there was a tuberculosis strain on the udder cows. So if they could isolate that, culture it some, make it what we call a virulent, not pathogenic, it might protect from human version tuberculosis. Sure enough, it does, okay? And it even protects cows from their own form of tuberculosis. But the story goes back a little further because as you start to see all this global clinical trial data starting to show the benefits of BCG, and not even starting, we're talking about phase 3 trials in Italy using BCG in new onset MS, better than the drugs we currently have approved. And then you see the trials in Australia using repeat BCG for allergies, okay? And you see the repeat trials of using BCG to prevent infections. You go like, what's going on here? Why is the reintroduction of this mycobacteria resetting the immune system? And it comes back to the hygiene hypothesis because as people have started to try to find more and more DNA samples from early humans and even Neanderthals, they keep coming up with BCG, effectively, mycobacteria in the bone marrow. So there's data now that, early humans 200,000 years ago even had mycobacteria DNA in their bone marrow at that time point. But if you look at modern man in North America, or modern women, I should say, right? Let's be equitable here. And it would take a sample of your bone marrow or my bone marrow today, we would find no mycobacteria in there. So mycobacteria is in the soil, right? But, I don't know about you, Ira, but this morning I wasn't out on the field.
I only do that on Sundays.
I did ride my bike, but I didn't go through any puddles, and I do buy my strawberries at Whole Foods, so they don't have dirt on them, and it goes on and on. So what's effectively happened is that some of these organisms that have co-evolved with us, literally, they were part of the cells in our body have been taken away. And when we take them away, because we don't have well water, we have antibiotics, and we have these clean houses, our genes are out of control.
Mm-hmm.
So when you got 200,000 years or 100,000 years of coevolution, the organism changes your genes and your genes change the organism, and there becomes a synergy, because these are smart bugs. Okay, these are not stupid bugs like Ebola. They kill you. Okay, these are bugs who want to live in you for the rest of your life. Okay, and when you take them away after that long of coevolution, you have unregulated genes. So that fits together very nicely, because often people with autoimmunity come in and they're trying to soothe their local water supply or find the industrial giant that contaminated their food or something. So it's not really what contaminated us, it's what we decontaminated that we like to think causes these really shifts in immune-mediated diseases.
You have obviously you have so much on your plate in terms of obviously diabetes. You have as mentioned, the entire basket of autoimmune disorders, which are in the United States alone, tens of millions. Overseas, you're into the hundreds of millions again. And then there's a third part of your, I guess, the triumvirate there in your lab, which is oncology. Now, BCG has this connection here in the U.S. with the bladder cancer product, which has made it's been in the press quite a bit lately with Merck and Sanofi. Well, Sanofi got out of its production because they don't like making it, but and so you have exactly. So you have one producer of it, but all that being said, that's not my question. We're in this era of these smart immunotherapies, these highly targeted products for hundreds of thousands of dollars, if not millions, which, they are doing some amazing things, but in very small populations of people. And I think once back, again, to sort of the oncology connection, that it seems like what you're working on in sort of the diversity of the responses that something simple like this can have. I mean, it seems like it could be a much sort of cheaper / broader approach potentially in oncology. Do you feel that there's a potential, in much broader than bladder cancer as it's currently used now to take a product this as well?
Yeah, I mean, that's a good question. I haven't thought about that much. I mean, the way, bladder cancer is kind of unique in that you can give, because it's in the bladder, although, the bladder wall is broken up from the tumor, you could give super high doses, right, without toxicity, massive doses. So BCG was advanced by other people and it's still used at some centers for metastatic melanoma, where they actually inject the BCG locally into the lesions to try to get single lesion disappearance. So there are some other oncology applications, but you have to think about not wanting to give a whole vat of BCG systemically because of the toxicity and the need for such high dosing. So I think that's what limits it a little bit in cancer.
Gotcha.
Although it's an extremely great drug for bladder cancer because of that containment. Yeah.
Now the question of the future plans, and obviously you're doing such amazing things the last couple of decades, but the future is still open. Obviously, I wish there was infinity dollars that could come down from the sky. But if there was, for you, where are you taking this the next 5-10 years? What do you see, as the further translation? Obviously, you're going to keep going with the diabetes. Are there other clinical trials that you have planned and some of the autoimmune disorders? Take us to 2030 and beyond.
Well, for BCG, obviously the multiple clinical trials we have going in type 1 diabetes are strong. They're going to get to a finish line, not 10 years from now, but much sooner than that.
Good.
And we hope we can keep the supply available for the public at affordable cost. So, this is kind of an interesting drug, right? I mean, it's old, really old. As you started out your program telling everybody it's like 100-year-old generic drug, and then I tell people it grows on the udders of cows. So, this is it kind of breaks the rules, right? Of simplicity. And I think, the applicability to other autoimmune diseases is just limited by dollars. So if the trials in MS, multiple sclerosis, are showing great promise in Italy. So if somebody came in with money, we'd certainly get trials going in multiple sclerosis with BCG. And then other trials will just take off based on the money supply. Like, somebody may say there's no drug for scleroderma. Why don't you try that? Or why don't you try it in rheumatoid arthritis? Or why don't you try it in psoriasis? Or, other allergic skin diseases? Can you smear it on the skin? I don't know. But as people start seeing the benefit of something affordable and safe, I think you'll see the expansion. And again, as you mentioned, it's limited by dollars to get these trials done properly and then the drugs approved.
Keeping count of time and obviously your schedule, just have a couple wrap-up questions that come back to you now. One just has to do with any influencers in your biomedical career to date that you might want to shout out to. Are there specific people that have kept you on this path over the years that if it wasn't for them, you would have gone into cardiology or done something totally different?
So one person comes to mind. Her name is Florence Hazeltine. So MD, PhD, who trained at MIT and then did clinical work in OBGYN and then ended up at NIH for years. And I think the thing that, she has so many lessons, not on how to do the science, because that I know, but how to do everything else but the science, okay? And was so, because, being older, even a tougher era for women in science, and understanding how science works and what to expect from science. And that wisdom along the way was invaluable. I mean, like things she'd say to me, I'd say, well, so-and-so doesn't like me. Because I'm female, I want to be liked, and she'd go, she'd say something like, Denise, do you like everybody? I go, no. Why do you expect everybody to like you? So just like good-hearted wisdom. Or, yeah, you can write that letter, but let me tell you what the reaction's going to be. And you go, okay, maybe I won't write that letter. Or, that's fundamentally wrong. This is what you have to do. This is how you should handle it. And this is how you move it up the ladder. So wisdom, I think a lot of people, and also just the concept of I'm mentoring you. Other people, as you get older, you should try to mentor people that work for you. Like learning the lesson that, part of being on this planet is not just doing for yourself or doing for your patients, but doing for your colleagues. And she was always a huge advocate of reminding people after savvy advice, in 20 years, you got to do the same for people who work for you. So I'd say just incredible. I'd like to use the word wisdom because when you get very well trained, you don't need somebody to say, Denise, you should have run this gel.
Right.
Denise, you should have designed the study with 20 more people. Or, Denise, you don't need that. You know how to do that. But dealing with infrastructures that are big and powerful and things like that, incredible wisdom. And so she gets a lot of credit.
Wonderful. Yeah. And then the final question, obviously, sitting there at Harvard, at Mass General, you've known, worked with, been mentored by a wide range of probably fascinating individuals over the years. This final question is about the person you did not ever get the chance to meet. And if I could take Dr. Denise Faustman in my time machine here and go meet someone at some other point in time that it doesn't have to be a scientist, by the way. It could be an artist, a philosopher, whomever. For Dr. Denise Faustman, who would that have been?
funny because my mind is somebody I quoted early, Mark Twain. I mean, a sense of humor. I mean, you got to have a sense of humor through all this. So, having at least 20 more one-liners to throw out when things get tense would be invaluable. Because here's a funny thing, Ira. My office is next to another department, and after being in this office for about 10 years, the department, kind of en masse, they're all in ministries, came back and they go like, "Why do we hear you laugh all day long? Like, what's going on in that office?" And I go like, bad things happen, and we just, we poke fun of them and we move on, like, can you believe this? No, we're going to top that, And so I think to keep moving in a forward direction, the gunfire on the sides just has to be ignored. And Mr. Iacocca, who supported our program for a long time, once said, well, they're not shooting at you. You must not have anything good. So realizing that they're only going to shoot and bring out the cannons if it's something that threatens their existence. So there's been gunfire along the way. But it's, if you think of it in the big picture, it's kind of inspiration that you're still ahead of them running. So
that's true.
I think that's how it works.
Dr. Faustman, it has really been a pleasure and honor having you join us today and share your background and everything you're doing with the audience. As we say on the show, this is all about sort of moving the human story forward, and you're definitely doing that. And thank you for that. And once again, everybody that's going to be listening, we've been talking with Dr. Denise Faustman, Associate Professor of Medicine at Harvard University, Director of the Immunobiology Laboratory at Mass General Hospital, and clearly doing some of the most fascinating work in both type 1 diabetes and other autoimmune diseases that you hear of. Follow her, keep an eye on the work, keep an eye on the clinical trials, because they are truly paradigm shifting, transformational, and really have the potential to change the dynamic in several areas of unmet medical need. So Dr. Faustman, once again, thank you so much for joining us. It's really been a pleasure.